Good afternoon, everyone. And I would like to thank both Prashant and Devendra for inviting me and giving this opportunity to talk to all of you about the work that we do and an exciting time that we all live in. The title of my talk is Genomics and Society, Role of Data and Common Sense. What I'm going to talk about today is how data has played a central role in our lives and how it all started, the birth of the field, what we call genomics today, and give you some examples where data has helped to understand biology better. And I'm going to give you three different examples from completely different fields, starting with the disease biology like cancer and understanding plant-based metabolites better, and then towards the end talk about how genomics can help to understand very interesting species in our biodiversity and understand about the biology about those species even better. Towards the end, I'm going to talk about why understanding data and the way we produce and share data openly is extremely important to our future, and how common sense plays a very important role. With that, let me start and take you back to the history. How did it all start? And of course, this is not inclusive of all the great discoveries in science that has taken us so far and has brought us to this date. These are some of the landmark discoveries from all the way back 1870 onwards. I'm not going to read all this to you. As you can see on the screen, right from the beginning, in the year 1871, from the discovery of nuclein by Frederick Mascher, and following the chromosomal theory by Sutton and Boveri, it all started when scientists took interest in understanding the genetics, or what we call the hereditary basis of life, and followed many different important discoveries like discovery of all the 4+1, if you take uracil nucleotides by Albert Cosell, for which he was given a Nobel Prize. And then the most important thing, even if you don't believe it, the term genome, which was coined way back in 1920 by a German botanist, Hans Winkler. In fact, people didn't really understand what genome was then, but the term was coined, and he used the term genome multiple times in the paper that he published then. The most important and the golden period of genomics started in the second part of last century, when Erwin Chargaff, came up with what we know as Chargaff's rules, both the first and the second parity rules, where he defined the nucleotide compositions in the DNA, and then the Hershey-Chase famous experiment where DNA for the first time was proven to be the genetic material, and of course the landmark discovery of Franklin Wilkins, Watson, and Crick when they discovered the double helix structure of DNA, followed by Nirenberg and Khurana's discovery of triplet DNA codon. If you go to the present, in the last 50 years, there are many different important discoveries in the field of genomics. And again, I'm not going to read this. As you can see, the most important one are related to sequencing of DNA, starting from Sanger, Wally Gilbert, and Paul Berg, both the DNA sequencing and recombinant DNA technology in 1970s, all the way to the invention of the instruments that are known today are the next generation sequencing and anything in between. Now, what has happened in the last 10 years is nothing but a miracle in the field of genomics, because the amount of data that we produce has really gone leaps and bounds, which is presenting newer difficulties and challenges to biologists. And we'll talk about some of those in the next few slides. Genomics as a discipline actually started, or rather coined, the term genomics is coined in 1987 by a scientist who worked on Tom Roderick, who worked in the Jackson Laboratory in Maine, which is the eastern coast of the US. And there actually started a completely new journal with the same name called Genomics. And in fact, if some of you are interested, there are very interesting anecdotal talks and newspaper articles about how this entire coining of the term genomics started in the local bar in close to the Jackson lab by Tom Roderick. The most important milestone, as all of you know, in the field of genomics started when there was an important effort to sequence human genome. And the project was coined then and came to be known as the Human Genome Project. The project traces its past to the Department of Energy in the US, and the effort was announced in 1986, but officially with the help of National Institutes of Health in 1990. As you all know, the draft genome was completed in 2000 with the landmark announcement in the White House by both the public and the private efforts, which was by the National Institute of Health as the public party and Celera Genomics as the private party. And the papers were published in the same time, both in Nature and Science in 2001, as the draft sequence and the complete sequence in 2003, which is exactly 50 years after the discovery of DNA as a double helix by Watson, Crick, Wilkins, and Franklin. Now, DNA sequences of human, what we know as the human genome sequence, is nothing short of a magnificent and unprecedented biological feat. Because can you imagine all that we work on today could have been possible without this sequence information? Hence, this is the resource, the magnificent resource which started many years back and we are using every single day. And therefore, towards the end, when I talk about data and why it is very important to produce data, good quality data, and deposit the data in the open domain so all of us can benefit, is an extremely important step. And this is just the newspaper cutting during the month of June 27, 2000, when the announcement came for the sequencing of the draft human genome for the first time. This is for the students in the audience, and this is a task for you to go back in history and identify these early pioneers. And all of them are pioneers in their own right, and every single individuals played a very important role in the field of genomics, and especially understanding human genome and doing the sequencing and analysis of the human genomes in early days. So it's a task for you, the younger audience, the younger crowd in the audience, to identify each of them and to know who they are and what their contributions are to the field of genomics. So I'm going to show you a few figures and take you through the last 25 years or so, how instrumentation or different innovation in the technology has helped us to sequence DNA better, faster, and cheaper. For example, this is a picture of 1994, how the early DNA sequencers looked like that helped us to sequence some of the genes during then. And then fast forward to 2006, these are some of the applied biosystems 3730, what we know, the automated DNA sequencer, which for the first time automated the whole process of sequencing, identifying the DNA sequences faster. And in fact, these are the instruments which are responsible for coming up with the draft human genome sequence in the year 2000 for the first time. And there are hundreds of them used both by the public as well as by the private effort. Now again, fast forwarding to 2020, as you can see, these are the Illumina HiSeq instruments, which helped for the first time to sequence the entire human genome of 6 gigabases or 3 gigabases haploid genome in just a matter of days. In fact, in less than 2 days. Again, if you fast forward today, we have the Illumina NovaSeq sequences. This is NovaSeq 6000, which helps to sequence multiple human genomes in the matter of hours. So as you can see, in the last 25 years, we not only have grown in technology, we have also grown how this technology has helped to produce genome data faster and cheaper and better, or with better quality. Now, This is a paper which is published in 2015 by Gene Robinson's lab. In fact, this is dated because this is, as you can see, 5 years old. But I wanted to tell you that this gives you an interesting idea about where we are going with the amount of data that has been produced by the field. This is a comparison of the data produced by the field of genomics with that from astrophysics and the common sites like Twitter, social networking sites like Twitter, and obviously very popular sites like YouTube. As the numbers say on your screen, the amount of data that is produced by the field genomics is already dwarfing those in other fields. Hence, the amount is a lot to deal with even today, and in going forward, we will have even complex problems to deal with the quantity of data. And towards the end, I'll tell you what are the solutions that the field or the scientists are thinking about to deal with this complexity of the data in addition to the amount of data. Now, this is a glimpse of where we are now in terms of the exact size of the data. There are multiple different databases today that exist that store human sequencing and non-human, for example, plants, bacteria, or virus sequences in the databases. And the largest one of them is the SRA, Sequence Read Archive database. As you can see on your screen, this data is, as of today, the 4th of December 2020, the amount of data, the total data, is nearing 46 petabytes, and it is growing very rapidly. Of course, this is from the one database. There are multiple different databases, and I have mentioned two other, the EMBL hosted database and also the Japanese databases, but this is by far the largest one. This gives you an idea about the humongous amount of data that is produced by the field genomics, including the data which is there in the database today. Now, as I have mentioned before, what are the drivers for this humongous amount of data generation? Now, access to instrument is one of the driver, because 25 years back we did not have those instruments that can produce data cheaper, faster, and better. We do now. In addition to the access, the cost, the low cost of the production of data also helped in producing a large amount of data. This has almost democratized the field of production of data, and you can find this next generation of DNA sequencers in almost all big labs around the world, which has helped to push the data quantity upward. In addition to the access and the cost, the efficient capture, storage, and sharing and analysis and visualization tools has also helped to get this data faster. Last but not the least, when the data is produced faster, we need a computational tools, technologies to catch up with those that produce data. And in the last 2 decades, the price to performance ratio for compute and other IT infrastructure has also caught up. Therefore, we have actually advanced quite a lot, not just in the data production, but also data storage, access, and analysis. And these are all the parameters that are helped, or rather the drivers that are helped to push the data production and storage and analysis at a much faster rate. But the most important question that we are going to deal with in the next few minutes are, is it useful to have this data? We have produced the data in a large quantity, but can we understand biology better? And I'll give you 3 different examples. The first one is from cancer, and our lab works on head and neck cancers to be specific, and I'm going to give you one example. This is actually different anatomical sites of the tumors of head and neck. We specifically work on tumors of oral cavity and multiple different sites on oral cavity, but today I'm going to talk about the tumors or the cancer of the oral tongue to be specific. Now, these are some of the pictures, as you can see on your screen, is the histopathology of the tumor. On the top, I am showing you three different images. The left one is just a white patch. It's not a cancerous lesion yet, but some of those white patches turn and become cancer at a later stage. And at the bottom, if you can see, these are the different stages. First, from a complete hyperplastic site to the site which has now become cancer. And one of the important question that we are asking with the help of data, can we understand which one of those patches turn into tumors? And if we do that, it will be much better to manage these patients with tumors better. We can identify the patients with the tumors before even the white patches become tumor. And I'll show you one particular question that we have asked and try to answer with the help of data. Now, as you will see in a minute or so, this has turned into quite an interdisciplinary science, because we want to understand this cancer at the level of the genome, at the level of the clinical manifestation of the representation, and also taking both the data together by doing or by utilizing the computational tools to understand and answer the questions that we start with. Therefore, we have a specific clinical question in mind, and we use different tools and technology encompassing all these different fields which is there on your screen to ask that particular clinical question and try to answer if you can do that with the help of data. Therefore, integration of data is very important, and integration of multiple different types of data and different dimensionalities in those data, and ask the question, can it help to answer the question that we start with? And as you will see in the next minute or so, we have been successful by using data from multiple different sources. First of all, producing data, high throughput data on these tumors, and collecting clinical data from those tumors and patients, and integrating those data with the help of machine learning tools to answer a very specific clinical question. And towards the end, this is an important point I'll reiterate. It is very, very important to start with a very specific question in mind and go back to answer that question with the help of data, if you are a bioinformatician or a computational biologist. Now, these are the papers that we have published, which essentially precedes before we ask the integration of the data. But first of all, we have to produce the data. So these are the papers which are cataloging different molecular determinants, starting with single nucleotide variants, mutations, different germline and somatic mutations, copy number variations, gene expression changes, methylation changes in these tumors. Now, once these data are produced, we actually took this data along with the clinical data and the epidemiological data to ask a very specific question. And in this case, we asked a question, can data help in identifying metastatic and recurrent tumors a priori? Now, what have we done? We have taken data on three different levels. Molecular data, as it suggests in the left-hand side column, with different types of molecular data, starting with somatic mutation, all the way to copy number variations and the context of that data in the genome. Then the clinical level data, the HPV or human papillomavirus infection in this data, the grade of the tumor, the stage, the node, and the habits, for example, whether they have tobacco positive or tobacco negative, and whether the tumor has actually recurred or not. And the third, the final data that we have taken is something which is already published on head and neck tumors, but not from our geography. For example, the cancer genome data, which is already deposited, the cosmic data, the International Cancer Genome Consortium data, and data from individual laboratories which have elucidated functional roles of some of those molecular determinants. Once we have these data together, we try to integrate them under a machine learning platform and a framework that I'm going to describe next. We call this machine learning platform or the framework as CAFÉ MOCHA, which is an integrated tool to analyze different type of molecular determinants to find clinically relevant signature. This is agnostic to the tumor type, although we have used this here to understand head and neck tumors, you can use it for any different tumor as long as you have the data and a specific clinical question in mind. This is a paper that we published in 2018 in Journal of Clinical Oncology, CCI, and if you are interested, you can go and read the paper. But very quickly, I'm going to show what exactly this paper we talk about. These are the different type of events and the molecular determinants we have taken. Some of those are categorical events like somatic mutation, some of those are quantitative events like gene expression, and some of them are functional events. For example, copy number changes where it is linked to its gene expression. As we know, if a gene copy is higher, we perhaps expect the gene to be expressed at a higher level. And if it is, we have taken that into consideration. And similarly, methylation changes linked with up or down gene expression changes which are linked to that. And once we have those data, we have devised a new, what we call as complete specificity margin-based clustering framework to understand that the relevance of a molecular determinant to ask a question related to the metastasis and recurrence of the tumor. In this case, I am showing in this figure 3 different stages. For example, whether it is a metastatic disease or a recurrent disease, or there is no metastatic or recurrent disease. And by each of the dot on your left-hand side is a single molecular event, where in this case expression, methylation, which are linked. And once I know that which one of those events are linked with a specific clinical determinant, I can actually use this particular clustering method. And I'm, due to time, not going to define or not going to go into the details. Some of you who are interested can go and read the paper. And once we know that which particular molecular determinants are specific to this event, we only select those. And therefore, we maintain a very high specificity. This is the final signature that we have obtained, which we call as MR44, which are basically 44 different molecular determinants for this particular type of cancer. And as you see at the end, we have validated that with an independent set of tumors, which gets a very high sensitivity. And because of the framework that we use, the clustering method that we use, we wanted to retain a very high specificity. And of course, as we get into more data, we will have different sensitivities in different type of data. So this is a beginning effort to understand whether we can integrate data to ask and answer a specific clinical question, and we have achieved that. Now, most important question that we want to answer, does integration matter? That means, is it important to integrate multiple different types of molecular determinants? And I'm going to show you in the next figure that yes, but please keep in mind, the importance of integrating multiple molecular determinants depends on the clinical question that you ask. In this case, I am obviously showing you from the 1 to 6. There are 6 different events that, as you saw previously, we are taking into consideration. And the height of the bar actually talks about the number of total molecular determinants which are responsible to give us the sensitivity. And the little black dot here talks about the actual sensitivity. Hence, in the last one, where we have a single combination, obviously when we take all the 6 different types, we get the highest sensitivity. Although if you reduce that to number 5, that means instead of taking 6 events, if I take 5 events, there will be 6 different combinations. And here it's not that bad, although it's not the highest, but we still get close to 75% of sensitivity. And obviously it goes down as I reduce the number of events. This gives you a glimpse that why integrating different molecular determinants is important. And as I said, it depends on the clinical question that you ask. Okay, so far, hopefully I have demonstrated that data helps to understand human diseases like cancer better, to actually answer a specific clinical question. Now, let's move on to the second example. The second example is from a completely different field, because the intent of the present talk is to let you know that data can help in biology. And in the second example, we are talking about a plant-based metabolite. Here we are talking about the plant, which is the neem plant. Almost all from India know about this plant because this plant is a very utilitarian plant. Through history, we have used different parts of the plant for multiple different purposes. And for today's talk, what is important is that let me go back one slide here, the important metabolites which are a part of the fruit of the plants. And there are more than 130 different secondary metabolites which are present in these fruits. And in today's discussion, I am only going to focus on a single metabolite, which is called azadirachtin, because this is an important, both agriculturally and pharmaceutically important metabolite, which is commercially exploited as well. Now, before we answer the question, can data help? We are the first group in the world to elucidate the genome and the transcriptome with a series of papers published. As you can see here on the screen, we did sequencing analysis and interpretation of genome and the transcriptome of this plant, and with the very important intent that we want to understand this pathways of this secondary metabolite biosynthesis better. Now, as I told you that the kernel of the seed of this particular fruit, neem fruit, is very important because it stores the secondary metabolite. Now, I'm not going to go through these particular slides. Basically, there are different classes of terpenoids, and the terpenoid that today we are talking about is azadirachtin, which is in the fruit. Then we started with a very specific question. And what is the question? The question is, can we use artificial intelligence, and in this case, specifically deep learning methods, to identify fruits and leaves with high metabolite content? And why is it important? It is important because the metabolite content in fruits varies between fruits and among different fruits of different region. And what we wanted to understand and work with is, can we use image-based deep learning methods to identify fruits and leaves which have very high metabolite content? In other words, can I just walk into a tree, take a picture of the fruit, and can say which fruit has higher amount of metabolite or not? Now, this was a serendipitous thought in our mind when we started it a few years back, and to our utter surprise, this method actually worked. Now, in order to do this project, we have traveled length and breadth of our country. So this is the orangish, figure that you see is basically the part that we have traveled to collect the neem fruits and leaves, which we have taken the images. And the blue southern states that we have done is that we have collected the fruits and leaves independently as a validation set. So this is again, published in this preprint bioarchive, and we are in the process of modifying it, getting it peer reviewed for the publication. So what is the premise here? The premise is that we have taken more than 80,000 images of fruits and leaves with the intent that the fruit images can actually be a surrogate to understand the intercellular concentration of azadirachtin. So we have two different inputs here. We have taken the image of the fruit and the leaf, and we also have biochemically, experimentally determined the metabolite content by using HPLC. So we have a HPLC derived actual concentration of the metabolite, then we have an image. So these are the part of the training set. And once we have that, we also have augmented the images, which has gone the part of the deep learning training set. Once we have done that, we ask the question, can we identify, or do the images represent what is there inside the fruit? And again, here I'm not interested to know the exact amount of azadirachtin, but I'm interested to know high, mid, and low. These are arbitrary, like cutoffs we have put up. So for example, I'm interested in anything which is more than X, I call it a high content. Anything between Y and X, mid. Anything below Y and Z, maybe low content. So that once I know whether it's a high content or low content, I can only select those trees for collection. And this is the result. To our utter surprise, when we used more than 83,000 fruits and 86,000 leaf images, we get a very high accuracy in terms of validation. As you can see, the 70% accuracy. Remember, this is the first time we have used this to use any images of the plant parts to understand the intercellular content class of this particular metabolite. As we speak now, we are refining this model further with more images and with more calculations, more computation to understand and to push this accuracy even further. Okay, so based on that, we have also made the desktop and the mobile app. And the idea here is that you can walk into a tree with your mobile phone, take a picture of the fruit, and instantly it will tell you whether the fruit has high or low content of the metabolite. And therefore, if you are interested in this metabolite, you can only choose those which has high content of metabolite. Therefore, it actually saves you a lot of energy, time, and effort not to go to the trees which has lower amount of metabolite. Okay, now hopefully I have demonstrated the use of data, a very different case in the plant. Now let's move to the final example. Again, a very, very different example. This is a nascent example, very early stage. We have actually just begun working on this, although we have demonstrated 3 years back the proof of concept. Now here, the use of genomics data to understand snake venom beta. The snake in question here is called Russell's viper. This is one of the venomous snake, beautiful animals, venomous snakes in our country. And there are more than, believe me or not, more than 1 million snake bite related deaths in India in the last 2 decades. There's a paper actually published in 2011 plus on neglected diseases, which talks about there are more than 60 different venomous snakes in India, but most snake bites, most if not all, are due to 4 different snakes: spectacle cobra, saw-scaled viper, Russell's viper, and common krait. And Russell's viper, the picture that you see here, is one of them. And the questions that we wanted to ask is very difficult to get permission to catch these snakes, because snakes belong to restrictive species. So it's actually prohibited to catch those snakes just for fun or even for research, because you need permission. So we wanted to ask the question, can we use the shed skin? Because snakes shed the skin periodically. Can we use this shed skin and get reliable amount, quality, and quantity of nucleic acid which can be used for sequencing? And once we use those for sequencing, can we actually do or get an acceptable quality of assembled genome? And finally, if we do that, can genome sequence be useful to understand the snake toxins? And I'll show you the proof of concept data for this paper, which we published actually in 2017. The answer to the first 3 questions is yes, and we don't know the last one, because we're still at a very early stage of understanding snake toxins, because it's a highly complicated cocktail of chemicals. Now, this is the paper that we published in 2017, which we have finally could use the shed skin to obtain reliable and high quality DNA and sequence the DNA, assemble this genome to get an acceptable label. As I'm using the term acceptable because the quality of the genome still could be much better by using multiple different technologies and by using multiple different tools to assemble those. But the quality is still acceptable to understand the toxins of this particular snake. Now, here I will just take a break, and so far I hope I have demonstrated the use of data in 3 different, completely different fields, but equally well how genomics and genomic data can be useful to understand biology better. Now, where does the field take us further? Now let's talk about a little bit on the data. What do we need to do about the data? Now, most important thing about data is that it has to be de-linked from the investigator. For example, if I am producing the data, you should not be, as a researcher, you should not be getting back to me and asking the question, can you give me the data? The data must be deposited in an open repository so that everybody can get it. There should be a common set of rules and the procedures, how to generate the data, how to share the data, and how to deposit the data. The data QC metrics has to be standardized, and the data, which goes without saying, must be shared ethically, must be shared equitably, and must take care into account if it is a human data, privacy, and we must have law and regulation that actually govern how, who, and when to share the data. Now, in addition to what I just talked about data, as you can see here, generation, access, sharing, security, findability, interpretability, and reusability of the data is very important. Therefore, we need to have a national policy. I'm very proud to say that I'm one of the few people who actually drafted India's first national biological data sharing policy, which is pending cabinet approval. And once it's approved, it will actually become a national policy which will govern how we share data with other researchers openly and in a responsible fashion. Of course, it goes hand in hand with the establishment of the National Biological Data Center, which is in progress, so that all the data which is produced can be actually housed in a common place and can be shared openly, equitably, and in a responsible way with all the researchers. And I'm not going to read this particular slide, but as you can see, we need to be putting our efforts how data is produced, which is there on the left hand column, and what are the different expertise that we need, which is the middle column, and what are the infrastructure that we need to ask questions. The important point here I want to mention that the bioinformatics expertise, because I'm expecting most of the audience in this talk are going to be from the field of bioinformatics and computational biology and genomics, is very important to distinguish that bioinformatics expertise not necessarily always equals to the expertise to deal with data in a responsible way. Therefore, we need to have training modules to train our future generation of scientists in this field, not just how to generate the data, but how to deal with the data in a responsible way. And I'm going to show you few solutions that I have in mind which can help doing that. Now comes to the common sense, and this is the part which is very important, particularly for the young researchers who are thinking of establishing their own labs in the field of bioinformatics, computational biology, and genomics. Now here is an important perception, and I'm sure that all of you face this some or the other time. And this perception, dare I say, is by most biologists, if not all. They think that the people who work with computers don't do any work, it's the computers who do the work. The real science is actually done by a biologist, and bioinformaticians are just a helping hand. They are just helping them to get from place A to place B, which is absolutely not true, because computational biologists or bioinformaticians actually do help more than help. They can generate their own hypothesis, and they can answer the biological question in a much more meaningful way by using data, as I showed you in the 3 different examples in my talk. Now, I have a very funny video to demonstrate what most biologists actually think, and I'm going to play that video to you. So here you see the guy in the black t-shirt is the biologist, and the train is the biological science. And the biologist is thinking that he's the one who is actually stopping the train, but actually what is happening, the train engine which is stopping the train, not him. And like stopping, it is again him who is going to think that he's the one who is going to start the train to go to the next level. Now he's trying to push the train. Little does he know that actually it's the engine which is pushing the train. This is of course a joke, but you get the point. Okay, now what is the solution? What can be the solution? All of you who is listening to this talk, is very important to be open. Do not ever be closed with your data. Put all your data in open domain. And you must always make sure before you try to publish anything, put your publication, the article, in a preprint server. You don't have anything to lose by doing that. You have everything to gain. Don't be afraid to work on tools only. Remember, tools are an essential part which will make sure that the discoveries are correct. Because if you work with the wrong tools, all your discoveries may turn out to be wrong. Work if possible with a question or a bunch of questions in mind, and as you will see in my next slide, stay away from the metrics. And one of the metrics which is overused not just in our country but all over the world is the use of journal impact factor, and you must stay away from this. I'm sorry for the haziness of the slide because it's probably just image which I've used from the publication. And here I'm showing you x-axis the retraction index and the y-axis is the impact factor. So more the journal impact factor, more is the retraction index, which means that the chance of getting the article or getting an article retracted is much higher if the journal impact factor is high. In fact, there are innumerable number of examples why the journal impact factor is one of the bogus, unscientific tools you can ever use, or metrics you can ever use to judge the quality of science in a particular paper. If you happen to be in the committee who is deciding, do read the paper yourself. That's the best way to find out whether the investigator has done good work or not. It may not be possible, I understand, in all cases due to time, but certainly not by using the journal impact factor as a surrogate. And similarly, the H index, stay away from this. They do not define who you are. That doesn't define the quality of science. What is rather going to define the quality of your science is the paper that you publish, how good it is, whether you have actually answered or tried to answer important biological question or have developed an important tool. Now, these are the last slides that I have. I can only tell you that the lessons that I have learned in the last 20 years working myself. You only work if you are passionate. That means you must be passionate in the subject that you're working with. Work with the questions in mind. Don't belittle the tool, as I mentioned before, because tools are very important. Give importance to quality over quantity. Okay, and not everything requires complex computing and doesn't require complex algorithm and large data. Sometimes small amount of data is also good enough to answer the question. Keep that in mind. Avoid jargons. Try to talk in simple language so that everybody can understand. Be humble. This is the most important point I want to tell you, especially the audience who are young people, students, young scientists who want to establish their own lab. Be humble. That's very, very important. Avoid focusing on where you publish and hence avoid the journal names, but focus on the quality. And as I mentioned before, avoid journal impact factors and H index. Enjoy science and the path of getting to the answers. Final slide. I would like to acknowledge the people who work in the lab, the hardworking students, the project fellows, the postdocs, funding agencies, collaborators in and out of the country, and many distractions. As you can see, I love Star Wars. So these are the two great characters that I like, both Yoda and Anakin Skywalker. Finally, the wise words, I'm sure you'll agree, of George Bernard Shaw, who told, "The reasonable man adapts himself to the world, the unreasonable one persists in trying to adapt the world to himself. Therefore, all progress depends on the unreasonable man." So, my request is don't be shy to be that unreasonable man. Thank you very much. And with that, I would actually love to take your questions.